Enzymes. By the end of this chapter you should be able to define catalyst as a substance that speeds up a chemical reaction and is not changed by the reaction. Define enzymes as proteins that function as biological catalysts. Investigate and describe the effects of temperature and of pH on enzyme activity. Explain enzyme action in terms of the lock and key hypothesis. What is an enzyme? A protein that can speed up the rate of metabolic reactions without itself being changed at the end of the reaction. They are also known as biological catalysts. Enzymes lower the minimum energy for a particular reaction to occur, thus speeding up the reaction. The minimum energy required to start a reaction is called the activation energy. The diagram shows the activation energy required for the reaction without the enzyme is higher as shown by the red line, and the activation energy is lowered significantly for the reaction with the enzyme as indicated by the blue line. A substance that speeds up the rate of a chemical reaction without being changed at the end. They are proteins in nature. They are needed in small amounts. They do not alter the end products of chemical reactions.
Most chemical reactions do not occur spontaneously in a cell. Instead, cells rely on proteins called enzymes to kickstart chemical reactions and speed them up, enabling cells to get the most out of the energy sources available to them. Enzymes have a unique way of kickstarting reactions. They work by binding to one or more specific molecules called reactants, or substrates. Binding occurs at a special region on the enzyme called the active site. Once the substrates bind to the active site, they form what's called an enzyme-substrate complex. As the enzyme and substrates begin to react, some of the chemical bonds in the substrates begin to weaken, causing them to link together. Eventually, the chemical reactions at the active site lead to the formation of a different molecule. This is referred to as the product. Once the reaction has occurred, the product is released from the active site. The enzyme returns to its original state and is free to react again with another set of substrates.
Metabolic reactions refers to all the chemical reactions within a living organism. Metabolism is equal to the anabolism or all the build-up reactions, plus catabolism or all the break-up reactions. A chemical reaction always involves changing one substance into another. Substrate is the substance which is present at the start of a reaction. Product is the substance which is present at the end of a reaction. The lock and key hypothesis. Each enzyme has its own active site which has exactly the right shape and size for the substrate to fit in. Enzymes are very specific. The lock and key hypothesis states that the enzyme acts as a lock and the substrate is the key, thus the only a specific key will fit the lock, active site of the enzyme. Substrate binds with the enzyme at the active site. Enzyme-substrate complex is formed. At this stage, enzyme breaks down the substrate. Products are formed. Products leave the enzyme. Enzyme is ready to bind with another substrate. The diagram summarizes the whole process. Many metabolic reactions are controlled by enzymes. Enzymes are vital in maintaining the normal functioning of an organism. It is more important than the air you breathe, the water you drink and the food you eat. Without enzymes, reactions would be so slow they would not occur properly. Enzymes play a critical role in digestion, germination of seeds, photosynthesis and many other vital functions. There are many types of enzymes, but here are 6 of them. Carbohydrase, catalase, lipase, protease, amylase and starch phosphorylase. You may have noticed that all the enzymes are being ended with ase. Effect of temperature and pH on enzymes. Being protein in nature, enzymes are very sensitive to temperature and pH. Like most chemical reactions, the rate of an enzyme-catalyzed reaction increases as the temperature is raised. But very high temperatures denature enzymes, as can be seen in the graph. The temperature at which the rate of reaction is highest is called the optimal temperature for that enzyme. Enzymes in the stomach have an optimum pH of about 2. Enzymes are sensitive to temperature because the enzyme and substrate molecules are constantly moving, using kinetic energy. The reaction only occurs when the enzyme and substrate molecules come into contact with each other. As the temperature is increased, the molecules gain more kinetic energy, so they move faster and there is a greater chance of collisions happening. Therefore the rate of reaction increases. When the temperature is raised beyond the optimal temperature, the rate of reaction decreases. This is because enzyme molecules are proteins. Protein molecules start to lose their shape at higher temperatures, so the active site becomes deformed. Substrate molecules cannot fit together with the enzyme, stopping the reaction. Denaturation is a permanent change in the shape of the enzyme molecule. Once it has happened, the enzyme will not work anymore, even if the temperature is reduced. An example of a protein denaturing is the cooking of egg white, made of the protein albumin. Raw egg white is liquid, transparent and colorless, while cooked egg is white. Changes in pH also alter an enzyme's shape. Different enzymes work best at different pH values. The optimum pH for an enzyme depends on where it normally works. For example, intestinal enzymes have an optimum pH of about 7.5. Enzymes in the stomach have an optimum pH of about 2. Extreme changes of pH may also denature some enzymes irreversibly. This is because the active site of the enzyme molecule can become deformed, as it does when exposed to high temperatures. As a result, the enzyme and substrate molecules no longer have complementary shapes and so will not fit together.